So for our last chapter, we're going to continue on with looking at the immune system. And in chapter 17, we looked at this side over here with the non-specific defenses, or the innate immunities that are built in, that includes things like those physical barriers on the outside of your body, the chemicals that are on the outside of your body. And then you work your way in and you run into what some people term the second line of defense, where you run into the cells and more chemicals and various actions that are occurring during this process of an infection or an invasion by a pathogen. And again, when we look at your immune system, the whole goal is to try and limit the spread of the pathogen and any toxic compounds it's producing, but also to clear the infection before it comes to cause too much damage. And one of the biggest problems with the non-specific defenses is because of the nature of them being non-specific, they are going to result in innocent bystanders being killed along with the pathogen. And so when we look in chapter 18 and we switch gears now, we're going to start looking at the specific defenses. And what we can see here from looking at this graph is really the key to what's known as the adaptive immunity that is going to give us these specific defenses. And the key to this is two things really. One is it has specificity built into it, and this is the part of your immune system that is going to target specific pathogens. And so I refer to it as, it's like putting a scope on a machine gun. Now, instead of just firing randomly, you're able to actually aim and specifically target the thing that is causing the immune response. And the other amazing thing about this is that you keep memory of that. So the cells that are responding during an infection or during this primary immune response after the initial exposure there, the second time that same pathogen comes around, you are going to remember what you've been previously exposed to. And note that the second immune response here and any ones after that are going to be faster and stronger after that first exposure, because you've maintained your memory of that specific kind of pathogen that was causing the infection. And when we look at this, you're going to run into a whole bunch of different terms associated with this stuff. So we'll try and go through them piece by piece here. But remember, in the previous chapter, when we were looking at our the white blood cells that are out there, they start out as a stem cell and they divide, and they can go on to take a variety of different roles depending on when they go through this process of hematopoiesis. And what we're going to focus on over here is these agranulocytes, the lymphocytes that are out there, and see what role they play in this adaptive immunity, and also how they're going to affect these cells over here that are the other parts of the complement of your white blood cells that are out there that are mounting that immune response. And the two major cells that we can look at associated with this adaptive immunity are your B lymphocytes. And we can see over here in the little cartoon, I attack invaders outside the cells. And these guys are going to be responsible for what's known as humoral immunity, because these elements of your immune system are in the body's humors or fluids, and they're going to attack extracellular pathogens and foreign objects that are found between cells. Likewise, if you have intracellular invaders, certain kinds of bacteria, different kinds of viruses that are out there can actually invade our cells, and we need to have some way of recognizing infected cells. And we also need to identify cancerous cells. And so those are going to be something that has a foreign object, but it's going to be on the inside of the cell. And so your T cells are going to be attacking infected and cancerous cells, and they're going to both boost your immune system, but also directly attack these kinds of pathogens that are found inside of your cells. And when we look at this process, we refer to foreign objects as antigens, and it just comes from the term antibody generator. These are immunogens that are generating an immune response, and they're typically going to be macromolecules that are out there. And if you remember our PAMPs, the pathogen-associated molecular patterns, and those are unique to specific pathogens that are out there. cells will have different receptors on the surface, different types of peptidoglycan, and so that's where we get the specificity from, because we can start to recognize those specific PAMPs that are out there. And so this whole cell right here would be referred to as an antigen because it does result in the generation of antibodies, but on that antigen itself, there are different molecules that are known as epitopes, and your epitope is a small exposed region of that antigen that can generate antibodies or generate an immune response that's out there. And when we look at good molecules that are out there that actually elicit a good immune response here, we can see complex molecules are really good. When you look at things like proteins, they are very complex with a whole bunch of different amino acids that are out there. So those are going to really elicit a good immune response. What we do find is repeating kinds of structures like these little guys here, those don't tend to elicit as good of an immune response. And there are some molecules out there that are known as haptens, and those are basically too small to actually generate an immune response by themselves. And what oftentimes happens with something, and a good example is penicillin by itself does not generate an allergy, but when penicillin sticks to cells in your body, now it's going to be a more complex kind of thing, and that is how people actually get antibody responses to something like penicillin that can result in an allergic response. So we're going to go through some of the steps in the adaptive immune response here. And I stole this figure from another book just because it shows you a nice little summary of what's going on here. And I've sort of artificially broken this down into a couple of discrete steps here. We'll look at the development of these lymphocytes and their maturation that we can see over here in this part. And then we will go on to the process where you do what's known as clonal selection and end up looking at the response of these lymphocytes, our B and T cells, to see what actions they actually have and effects on your immune system that's out there. So if we go through and look at this process here and look at this lymphocyte development, remember we're looking at these cells that are going through this process as they start to develop in the bone marrow and the thymus. Your B cells are actually named after the bursa in the chicken, where these guys are going to go through maturation, and the T cells get their name from the thymus. And one thing I should point out is that your T cells are really active in your younger years. As you get older and older, your thymus actually will fill up full of fat cells. And this is why one of the reasons why they include thymus and chorizo, because it gives you that nice little fatty addition to your sausage there, along with salivary glands and whatever kind of lymph nodes that are left over. Those will all go in to make that nice little tasty breakfast addition there. But when we look at this process, these receptors that we see here are actually embedded in the plasma membrane of these two types of cells here. And the receptors are glycoproteins, so they are actually 4 chains for our B receptors of these proteins that are held together by these covalent disulfide bridges. And when you look at your T cell receptor over here, it's actually got 2 chains that are embedded in the membrane there, held together by a disulfide bond right there. Notice on both of these guys, there is what's termed an antigen binding site here. And that's going to be important because the changes in these receptors right here are actually going to occur as these lymphocytes are going through that development process right there. They will actually use about 500 different segments in some genes that will actively recombine. So the DNA is literally being mutated on purpose by these lymphocytes to generate different coding regions within the gene. And so when you look at these variable regions right here, they're actually produced randomly by the cells as they go through this process. And once they go through that recombination where they mutate these variable regions, that gene is locked in place right there. And so now you've got a B or a T cell with a receptor sticking off, and each B or T cell is making a slightly different receptor every single day of your life. So it's important to realize these are randomly generated, and they are not formed in response to the presence of an antigen. So when you go in and get vaccinated, there is a chance that you didn't happen to make any receptors that bind to the vaccine particles that were injected into you, and that would mean that you did not develop a memory of that particular antigen that got inside of you. So every day of your life, you're going to be producing new B and T cells. And if you think about it, if we're producing more and more of these, eventually we're just going to be a big old sack of lymphocytes. But these cells are going to go through some changes right there. And eventually, when they do mature, you can see from this little picture here, they've got these little receptors sticking off the surface. And to give you an idea, they estimate that a typical B cell is going to have 500,000 of these receptors sticking off the surface. So you can think of this as just a cell with all these hands sticking off of that, and each hand that is on the surface is only going to be programmed, or only is programmed to bind to one particular type of molecule that's out there. And so we want to go through this process and take a look at, now that you've developed this lymphocyte here, it's in your bone marrow or it's in your thymus, and if you're making hundreds of millions of these lymphocytes every day, you need to get rid of the ones that actually don't work properly, and that's where we go through this process of maturation. And one part of that is what's known as clonal deletion, and this is really where those B cell and T cell receptors are actually going to be tested. And if you think about it, you've got all of these cells out there, each of them with a unique receptor on the surface. Well, some of those receptors are going to be able to bind to self. We don't want those cells going out into circulation, so those cells are going to undergo what's known as programmed cell death, or apoptosis right here. So if you stick to me, I want to get rid of you before I send you out to patrol my neighborhoods. And so we get rid of these cells with receptors that bind to self as these lymphocytes are going through this maturation process. You've also got cells that just have messed up receptors. Again, it's a mutation, totally random. So some of them just aren't going to function well. Those cells are also going to undergo apoptosis and go through this programmed cell death. If you do have a good receptor that doesn't trigger either one of those things, they're going to continue on through this process. And when we look at this clonal deletion is going to give us what's known as immune tolerance. We can tolerate ourselves because we've gotten rid of the things that bind to us. And when they do some estimates on these populations, they think that in your thymus, if you're making, say, 50 million B cells on that day, you actually are going to eliminate about 98% of those. So a huge repertoire of these cells never actually makes it past this clonal deletion step. But then we've got these last guys here, the ones with the appropriate receptors that aren't activated by either one of these steps. They're going to continue on through this process, and they'll go through what's known as clonal selection. And now we take those lymphocytes and they are going to go into circulation. They will head out to the bloodstream and they will go into lymphatic tissues. And it's important to remember each of the cells has this random variable region on that receptor, whether it's a B cell or a T cell. And cells that are floating around out there that actually are going to bind to a specific antigen, those guys are going to get activated to divide. And when those cells divide, we get clones of the original one. And so this is known as clonal selection, because we're selecting out of all of those hundreds of thousands of cells that are made. A bunch of them are deleted, but now we've got ones that are going into circulation. And if they stick to a foreign particle, that is a signal for the body, or for those cells, to divide by mitosis and make copies of themselves. And each of the copies is going to have the same receptor on the surface that was made as that lymphocyte went through that maturation process right there. And so now we've got a clone or a pool of all of these cells with the same receptor. And some of those cells are going to get saved as memory, and some of them are going to be used as what are known as effector cells that are out there. And we'll take a look at what happens with this process in the next videos.